Okay, better. So, yeah, thank you for the introduction. And I'd like to appreciate this opportunity from NCCEH and thank you for joining me, everyone, at this very busy time of the year. So, as Tina has mentioned, the topic of my talk today is about the multi-drug resistant pathogens in foods. And my lab at UBC has an emphasis on using molecular approaches to understand the microbial pathogens. Many of them I will actually mention today, whether regarding how they are posing threats to food safety, food security, and public health. I just want to briefly do my own land acknowledgement, and we thank for the Musqueam people for maintaining this really beautiful land for us to conduct our research activities on. So with regarding antimicrobial resistance, I'm pretty sure that is a topic that's actually very close to our hearts. We know that it actually concerns so many different sectors that we're basically experiencing on a day-to-day manner. And with regarding what I will be focusing on today, which is the multi-drug resistant pathogens in foods, I will be particularly focusing, as you can probably imagine, on the form side of the story. And this would include the forms about raised animals for human consumption, as well as the farms for growing the crops, vegetables, fruits. Aquaculture is actually also another very important sector to consider. But for today's topic, I won't be spending too much time on aquaculture just because I really want to focus on what is happening in Canada and what are the most pressing issues to us. Whether regarding aquaculture, it is indeed a hotspot for multi-drug resistant pathogens in other parts of the world, but it's not as a big issue in Canada. And because the topic itself covers such a wide area, I really want to focus on what's happening in Canada. And these foreign environments, as I just mentioned, as we know, they are closely tied to the natural environment in general, which is on the center of this picture. So we're talking about the lakes, rivers, and the soils, which are closely involved in, well, the food production as well as potentially harboring these organisms that we are concerned with in terms of causing issues to human health. So that really reminds us how the natural environment is important to us. And when we're dealing with issues related to antimicrobial resistance, we do have to take a holistic approach to look at these issues. So further actually zooming into some of the specific routes with regarding how these multi-drug resistant
Sorry?
pathogens can enter into our
무서워요.
food systems. So it's a really
무서워.
complicated picture with regarding, well, as we can see, multiple players involved. So food producing animals can develop drug-resistant bacteria in their gut and get further passed into the meats and other food products such as dairy that we will consume. So this is probably the most intuitive way that people have been thinking about, but we also have to pay attention to the fact that these multi-drug resistant pathogens can also be passed to other factors that are involved in the environment, such as soil and water, which further impact the food crops. And eventually, consumers can get these pathogens through the consumption of food if these foods have been contaminated and therefore That's how harm can be done, actually, in terms of causing the diseases associated with these drug-resistant pathogens. So I mentioned that I would really like to focus on what's happening in Canada today, and that's why I will run through basically a review with regarding some of the pressing issues associated with different food producing sectors. I'll probably spend a bit more time on the poultry sector just because we do see some issues that are associated with the poultry sector in terms of multi-drug resistant pathogens. I will also be covering the beef as well as the pork and the food crops, such as fresh produce. So I will start by sharing a little bit about what's going on in the poultry sector. So And I tried to, when I was preparing for this talk, I tried to actually find examples that are relevant to Canada. So these studies were either conducted in Canada or maybe they have some relevant elements. So this was actually a survey done throughout the period of 2013 to 2019. And it's a very interesting study because it actually correlates the elimination of the preventive use of different categories of antibiotics. So we know for category one antibiotics, they were actually pulled off from the poultry sector back in 2014, and then in 2018, we eliminated the use of the category 2 antimicrobials. And then later in 2020, which was actually not reflected in this paper, there was the elimination of the use of the category 3 antimicrobials. So, as we can see among these three very important foodborne pathogens, Salmonella, E. coli, and Campylobacter, with the elimination of the use of these certain categories of antimicrobials, we are also seeing overall a trend of decreased mean proportion of resistance among these pathogens to these antimicrobials, which is totally understandable, and that's why people are doing it. But there's also another side of the story. So, because people were not using the category 1 and 2 antimicrobials, as a result, we're seeing that for some pathogens, such as Salmonella, if we're paying attention to what's going on uh, with the mean proportion resistance to a category 3 antimicrobials, such as tetracycline, there is some increase in the resistance of these category 3 antimicrobials. That also makes sense because if people cannot use the other categories anymore, they will go to the category that's still being allowed, right? So that actually reminds us when we're taking these strategies with regarding reducing the use of antimicrobial resistance, antimicrobials, there is also potentially consequence related to the rising resistance associated with the antimicrobials that are still allowed. So that's one side of the story. I would also like to share another side of the story. So this study was actually done on commercial broiler chicken farms in Quebec. So the researchers basically found that yes, it was great to withdraw the use of antibiotics, but there were other consequences. Aside from the rising resistance I just mentioned, it can also have other implications, such as, well, the intestinal microbiota composition of these broiler chickens can change. Well, it can go either way. It can be positive, but in this case, it wasn't really positive outcome. The researchers have found that some of those beneficial microorganisms, as well as prebiotics, uh, like the prebiotics they're producing in terms of fostering the well-being of the intestinal microbiota of these broiler chickens, are reducing as a consequence. So in another word, by doing the antimicrobial withdrawal, um, it is potentially harming the well-being of these broiler chickens. Another consequence is, of course, the production performance decreased. And some harmful microorganisms, such as Clostridium perfringens, otherwise have an increase in their populations. So this is again telling us there are some consequences with the antibiotic withdrawal, and some of them can potentially harm either the animal health or maybe fostering the growth of unwanted microorganisms. Another aspect I would like to touch on is actually food safety. Actually, the consideration of antimicrobial resistance and the occurrence of foodborne pathogens in foods that can potentially harm human health, although these two topics are very much related, they are not the same. So, in another word, even if we're seeing overall a reduction of antimicrobial resistance, that doesn't mean essentially we're seeing a reduction of the occurrence of these pathogens. So this was actually data published by Public Health Agency of Canada back in 2020 regarding the percentage of retail chicken breast samples positive for Campylobacter, which is a very prevalent foodborne pathogen in uh, chicken overall. So as we can see across these different sites that they have surveyed on throughout the years, that correlate with the withdrawal of the category 1 and category 2 antibiotics, the percent of samples positive for Campylobacter didn't actually experience a significant change. Actually depends on the province that we're seeing. Sometimes they just stay very steadily across the same level, or maybe they decreased a little bit and then rise back. So overall, the strategies that were taken in terms of reducing the usage of antibiotics in chicken farming didn't really help with reducing the occurrence of foodborne pathogens. And that also reminds us uh, other strategies have to be taken in terms of how we can effectively manage the occurrence of foodborne pathogens in these animals and further down on the food products that come from these animals. And then now that the options of using antibiotics are out of the picture, we actually have to come up with some new ideas. And at the end of this talk today, I will very briefly introducing some of these new ideas. So that was the overall picture with regarding what's happening in the poultry sector. So the next slide or two, I will be briefly talking about what's happening in beef. So, with regarding the overall monitoring of antimicrobial resistance in livestock and the retail meat, it's the job of the Canadian Integrated Program for Antimicrobial Resistance Surveillance, or CPARS. So, if we go to their website, we can actually find a lot of very important and useful information. So, compared to the poultry sector, the AMR, antimicrobial resistance, and multi-drug resistant pathogens are actually at relatively low occurrence and are not increasing. So in another word, it's not as big as an issue. Because the drugs of very high importance are actually very rarely used in beef cattle in Canada. So here is one example with regarding the extended spectrum beta lactamase producing E. coli or ESBL, E. coli for short. So we know that these pathogens that can produce beta lactamase can be a big issue because essentially that's how they can do in terms of getting resistant to the treatment of antibiotics such as penicillin. So in this case, the CPARS reported their surveillance results with regarding different species of animals. As we can see, there was chicken and turkey and pigs. And at the retail level, there was chicken, turkey, beef, and pork. So across different farm to food supply chains, the different points, they actually tested a lot of samples. And overall, what we can see is that from the chicken meat or from the chickens on farm is where the E. coli recovery rate was the highest. And the same with the number of ESBL E. coli and the prevalence of ESBL E. coli as well. So that study, although it was only on the ESBL E. coli, it also provides important information with regarding how these antimicrobial resistant pathogens are actually impacting different food sectors. So overall, it seemed that chicken and turkey are the ones most impacted. And in beef and pork, yes, we do also see some occurrence, but maybe not as much as in chicken and turkey. And that actually serves a really good reminder to us because in the upcoming holiday season, we are gonna eat a lot of turkey, and it's always important to actually keep in mind that when we're cooking something like a turkey, it's important for the internal temperature to reach 165 degrees Fahrenheit or 74 degrees Celsius. Um, Although we're only looking at E. coli here, other pathogens such as Salmonella and Campylobacter can be carried by turkey and therefore potentially harm the people who are eating them if they're not effectively got rid of. Okay, so this study was actually done in the maritime provinces. And it was an AMR profile on extended spectrum cephalosporins resistant E. coli isolates recovered from retail meat products. So instead of surveying everything from farm to food, now we're kind of zooming in the picture to focus really on what's happening at the retail level. So basically, what the consumers are getting from the grocery stores. So, as we can tell here, the numbers are actually pretty much telling a very similar story to the previous chart. So we're seeing that there were actually isolates of AMR E. coli being recovered. There were more in terms of percentage from chicken and turkey than from beef and pork. And the analysis was actually done with regarding how they're resistant to different antimicrobials as provided in the list. And as we can tell here, so the most prominent ones are probably like ampicillin, as well as the other ones on the top, but also they can potentially be resistant to the other antimicrobials that are very important for clinical treatments. Um, and then that's a good reminder again to us because if, so say, someone actually eats the meat that was contaminated by these uh, antibiotic resistant pathogens, it will potentially be harder to treat their infections because the um, choice of antibiotics will be pretty much limited based on what these pathogens are resistant to. Okay, the last um, animal derived product I would like to mention is actually pork. So, with regarding pork, other than what I have already gone through in the previous two slides, I would also like to briefly mention about another pathogen, which is also pretty familiar to us, the methicillin-resistant Staphylococcus aureus or MRSA. So, a survey was done in Canada, but quite a few years ago. I wasn't able to find a more updated study, but maybe there is one somewhere being conducted. So, in this survey across four Canadian provinces, BC, Saskatchewan, Ontario, and Quebec, essentially MRSA was found in pork samples at retail stores across all of these provinces. So, at the same time, I found a study which was not actually conducted in Canada. It was done in Europe, but I personally found it to be very interesting and alarming as well. So, essentially, in Europe, people have found that there has been a highly antibiotic resistant strain of MRSA. This particular strain is referred as CC398. It basically emerged in the livestock in the last 50 years, and it is still there causing issues. So essentially, in the past half of a century or so, the same MRSA strain has been causing issues with regarding introducing the AMR in the swine and pork sector in Europe. And it's probably due to the widespread antibiotic use in pig farming. So researchers have done quite extensive whole genome sequencing based phylogenetic analysis, as we can see from these charts that I will actually not get into the details. But essentially, what the researchers have found is that there's a very stable resistance associated mobile genetic elements that are presenting in this MRSA strain. And essentially, this mobile genetic element has been contributing to the resistance of the MRSA strain. And therefore, the impact or the consequence is even though there has been reduced use um, in antibiotics in European farms on livestock associated MRSA, it will be very slow to be realized. So typically, we're hoping that by reducing or withdrawing the use of antibiotics, we will be able to see the corresponding decrease of antimicrobial resistance in that sector in the next few years or the foreseeable future. But the story is more complicated than that. As we can see, the strain has been hanging in there for the last 50 years. It most certainly has some genetic traits that would allow it to pass the resistance just over and over. And then that's why by reducing the use of antibiotics, it won't simply result in the reduction of antimicrobial resistance on farm that easily. Okay, so now I would like to actually move on to the crops. So the food crops, which I would assume that it can run everywhere from vegetables or pulses and grains or herbs or salad crops, but especially according to the studies have conducted so far, um, including this review paper, which actually conducted a really nice meta-analysis of the studies published between 2000 and 2020. As we can see, if the color is like in a darker blue, that means there are more observed sample groups associated with the antimicrobial resistance with that particular pathogen. And what is the pathogen that we're seeing the most of issues? It's E. coli. We see a lot of darker blue spots associated with E. coli across different food crops, but especially in vegetables and salad crops, where we're seeing actually the darkest color, which indicate the most observed sample groups. And E. coli. Following E. coli is Salmonella. It's another very important pathogen that is actually the number one cause of foodborne illnesses across the world. And the fact that they're causing so many issues or so many contamination events in vegetables and the salad crops uh, is actually pretty alarming. Because one major difference between now that we're talking about these food products compared to uh, the meats is that for meats, one important message we are always repeating over and over to the consumers is to cook them properly. But when something like salad crops are consumed, they are not cooked. Like people would not cook their lettuce or cucumbers under just normal circumstances, right? So that is why over the past few years, we're just seeing an increasing number of foodborne outbreaks associated with these pathogen contaminations that can be linked to fresh produce items. There are several reasons. The first is people nowadays, we actually prefer fresh and minimally processed food products that are nutritious. And these products, such as salad crops, are providing a very ideal option. But at the same time, as I just mentioned, there is no cooking step, which is critical for eliminating these pathogens that is being applied to these vegetables. And that is why we are seeing quite a few outbreaks. Of course, many of us still remember the outbreak of E. coli infections linked to romaine lettuce in the past few years. Actually, there has been more than one. And most recently, the US FDA actually issued an announcement with regarding outbreak investigation of salmonella that was contaminating the cucumbers. And that was just taking place back in November. So the investigation is still going on, as we can see. All these events are actually reminding us how important it is to make sure that our fresh produce are safe from the contamination of these foodborne pathogens. So for today's talk, I have been mostly focusing on basically my literature review findings. But at this moment, I do want to share a little bit with regarding our own research, because it's also relevant to the topic with regarding fresh produce safety. So a few years ago, we actually conducted a survey with regarding the irrigation water safety in the lower mainland of BC. So essentially, PhD student Justin, he went out to the Fraser Valley, which was a very important region for agricultural production in BC. And he took irrigation water samples Irrigation water, by the way, is one of the major causes of pathogen contamination on food crops. As you can imagine, if the water is contaminated, the pathogen can be transferred from water to the crops potentially. So essentially, we tested for uh, cytotoxin producing E. coli, Listeria, and Salmonella, which are the three very important pathogens associated with that fresh produce. And we essentially found that, yes, there were pathogens in the irrigation water, and depends on which pathogen we're talking about. Sometimes there were more being collected from um, a certain category, like overall, we found more Listeria monogenous than E. coli or Salmonella. We did find actually quite a few E. coli isolates, which can be pretty alarming uh, based on what I have just discussed about how it is impacting, well, the produce safety and responsible for these outbreaks associated with lettuce and other fresh produce. We also did further studies into how these E. coli isolates are like. Are they actually multi-drug resistant? So this work was actually done by another grad student in my lab, Yvonne, and she evaluated the resistance of these pathogens and did find that almost 15% of these isolates were multi-drug resistant. So now that we know, yes, there are pathogens in the irrigation water, and some of them are actually multi-drug resistant and can potentially pass their resistance to the crops that people can potentially consume. And if people consumed these crops that are contaminated with these multi-drug resistant pathogens, then again, I mentioned this before already, it will be harder actually to treat these infections because of reduced options of antibiotics to be used. Okay, so that was what I would like to talk about with regarding AMR in food crops. So far, we have gone through examples with regarding AMR in the poultry sector, in um, beef, in pork, and in food crops. So to wrap up what we're actually discussing about today, I would like to very briefly introduce some alternative antimicrobial strategies because I did mention earlier now that the use of antibiotics is out of the picture. For many of them, what are some of the other strategies we can actually use to reduce the foodborne pathogen contamination across different food sectors? So if we actually look up literatures, papers, books, you name it, we will find there are quite a few different alternative strategies, such as the use of probiotics and prebiotics. And this one is probably one of the most familiar to us. And the use of antimicrobial peptides and bacteriophages, I will actually get into some details with regarding these two because there are some interesting facts with regarding these strategies. Organic acids, which are very typically used during food processing. Essential oils have a lot of potential, but we also have to pay attention to, well, the fact that many of them are very fragrant and that they can change the taste and the smell of food products and small molecules. So for each of them, I can basically spend another hour just talking about them. But considering the time constraint, I will be briefly introducing the antimicrobials as well as the bacteriophages. I will start by talking a little bit about antimicrobial peptides. So antimicrobial peptides are actually naturally occurring and they can come from many different sources. They can come from animals and then they can also come from bacteria. And the reason that I'm using bacteriocins as the example here is they're probably the most successfully commercialized antimicrobial peptide products in food industry. So what are bacteriocins? They're essentially uh, synthesized by the ribosome of bacteria. Some interesting history regarding how they were discovered. That was actually back in the 1928. That was same year that Alexander Fleming discovered penicillin. So researchers have found actually during the cheese making that certain lactic streptococci strains were able to inhibit the starter cheese cultures. That wasn't actually ideal in terms of cheese making, but that's a very interesting fact because these lactic streptococci strains are genetically very similar to the starter cheese cultures. They can all be considered as lactic acid bacteria. So that is one important fact about bacteriocins. They are actually produced by certain bacterial strains for the purpose of fighting against their closely related relatives, like their cousins, in terms of competing for the limited resources in whatever environment they're in. Why is that important? Because that pretty much is telling us the antimicrobial spectrum of bacteriocins is way narrower compared to uh, antibiotics such as penicillin. And why is that important? Because one of the key considerations for penicillin and other antibiotics is that because they have such a wide spectrum, they will wipe out most of the members in a microbial community. And essentially, that's how AMR rises. But because bacteriocins are having such a narrow spectrum, then in that aspect, we don't have to worry as much about them wiping out the entire microbial community, which also includes many beneficial members. So one most prominent example of bacteriocins that is commercialized and being widely used in food industry is actually niacin. So we already talked about that. It was first discovered in 1928. And throughout the years, as we can see, that's how niacin is used as a food biopreservative. And nowadays, we're seeing more and more biomedical applications of niacin. So, um, that is, well, the positive aspect about niacin. There are also some challenges. For example, similar to developing AMR, essentially bacteria can develop resistance to antimicrobial peptides such as niacin as well. There are many, many mechanisms involved, but that can potentially happen, and that's why we need to be aware of that, the development of bacterial resistance to AMPs. And the production cost is in general a bit high just because fermentation is involved. And of course, we have to use big bioreactors for that purposes. And then there are also some regulatory considerations. We're seeing a lot of bacteriocins or antimicrobial peptides being evaluated, and many of them showing very promising applications, but only very few are actually approved to be used in the agri-food sector. So the next one I would like to briefly mention about is the bacteriophages. So in terms of the discovery of the bacteriophages, it took place in the also in the early 20th century. So for that era, it was actually a very fun time for microbiologists. So it was a British microbiologist and a Canadian, French Canadian microbiologist. And both of them actually sort of discovered bacteriophages around the same time. So essentially, when they were culturing bacterial cells on an agar plate, what they found was some parts of the agar plate actually became transparent as if something was eating them up. And turned out that these were the bacteriophages that were actually eating the bacterial cells because bacteriophage literally means bacteria eater. So um, on the lower part of the slide is how a bacteriophage actually looks like. It's a bit um, it has a bit alien look, and the head is actually where the genetic material is. And then these tail fibers are also very important because they actually use the tail fibers to and associated receptor binding proteins to interact with the host. That's how they notice this is a bacterial host that they can actually invade and infect. So these tail fibers are actually very specific. For example, if we're talking about a bacteriophage that can infect Salmonella, typically it won't actually be able to infect other bacterial species that are very closely related to Salmonella. So what is that implication? It also is telling us they have a much narrower antimicrobial spectrum compared to antibiotics such as penicillin. And for that reason, they have the same benefit as the bacteriocins we just went through. Challenges with regarding the application of bacteriophages? Well, they can also result in the bacterial cells developing resistance to phages. So essentially, when we're thinking about bacteria, they're very smart. They can develop resistance to all these antimicrobials that we've been talking about so far. So you're probably getting a bit frustrated. Like, what do we do then? I will actually talk more about that in the next slide. They're very, very specific to their hosts. So sometimes one salmonella phage won't even be able to infect all the different salmonella strains. And then that's why when phages are being used, as far as the currently commercially available products available, um, I would say most of them would involve the use of more than one phage. It will be in the form of cocktail, just so that they can have a wide enough host specificity across different strains within one species. And there are some regulatory considerations associated with the approval of the use of these phages as well. Okay, so my last slide before I wrap up, I did mention that like bacteria are able to actually develop resistance against antimicrobial peptides as well as bacteriophages, which just seems to be a bit frustrating. Like there's no one solution that would fit all. But it's important to acknowledge that having a wide selection of repertoire with regarding all these choices do help us with developing more effective strategies to combat with these antimicrobial resistant pathogens. So one example is that bacteriophages can be used when the multidrug-resistant bacterium is doing harm to people. In this case, we can see in the very pretty recent news that they're being used to fight against a multidrug-resistant Klebsiella pneumoniae. So in these cases, if the regular antibiotics are no longer working, we would still have other options. Another side of the story, yes, bacteria and bacteriophages or antimicrobial peptides, they are actually having this constant battle against each other. It's like a molecular arms race. Bacteria would try to mutate in order to develop resistance. But one thing we have to acknowledge is they never become invincible. In another word, when they're trying to mutate to gain some kind of resistance against one agent, chances they have to make sacrifices. There is always a trade-off. They become stronger in one aspect. At the same time, they will become weaker in other aspects. On the right-hand side, this is a graph actually showing from one study on multidrug-resistant Pseudomonas aeruginosa strains. When they are trying to evade uh, being killed by bacteriophages, they actually have to mutate in a way that makes them more vulnerable to other drugs, including antibiotics. So, in another word, we can potentially use this as a strategy to revive the antimicrobial resistance, the vulnerability of to antibiotics across these multi-drug resistant pathogens. So when they are to evade evade bacteriophages or antimicrobial peptides, they can potentially become more vulnerable to the antibiotics that they used to be able to fight against. So this is very complicated dynamics, and that's why more research is needed to understand how these bacteria will actually evolve and potentially how we can manipulate this process to actually drive them towards somewhere that they will become vulnerable to antibiotics again. Okay, so to close my talk today, I just want to say that when we are talking about what's happening in the agri-food sector, we can never forget how important it is to keep a holistic approach to pay attention to a healthy ecosystem and healthy animals and healthy humans all together. So that's why the One Health concept is very important. So for the research parts that I presented from our lab today, I would like to thank our founders as well as the talented students who did the work. And of course, thank you all for listening to what I have been talking about here. So that will be the end.